Hi, my name is Éloi Durant and I'm a PhD student at the University of Montpellier in France. I'm working on the visualization of pangenomes, and today, with my poster A Look at Traits Through the Pangenome Visualization Jungle, I wish to give you an overview of the field and introduce a dedicated tool I developed. First, what is a pangenome? A pangenome is an inventory of genomic material within a group of related individuals. Let's give some examples. The first definition was applied by Tetler et al. On bacteria. They were studying the genes and discovered that some of the genes were absent from some of the strains. They defined different categories, the core genome and dispensable genome, the core genome being all the genes that were present in all of the strains, while the dispensable genome was the genes that were absent in at least one of them. Combined together, the core genome and dispensable genome constituted the pangenome. This definition has been extended to fit eukaryotic species with much more intergenic space. Their genomes can be considered as successions of chunks of sequences, and these chunks of sequences can be put together within a graph. Each genome is therefore a path through that graph. This definition of pangenome therefore evolved from a gene-based pangenome definition to a sequence-based definition. For the visualization of these definitions, multiple representations has been proposed. We can divide them between multiple categories. The unspecific and gene sets representations are mostly fit for gene-based pangenomes, while the positioned and structural representations are mostly fit for sequence-based pangenomes. Multiple tools have been created for these representations. However, none of them really suited our need for plant pangenomics, either because they were dedicated to genes pangenomes or because they couldn't scale the size of plant pangenomes. We therefore worked on new representations and a visualization tool. This tool that I would like to introduce today is Panash. Panash stands for Pangenome Analyzer with Chromosomal Exploration. Our take for this representation is that the genomes can be segmented into different pangenomic blocks, and those blocks can then be laid out on a single thread, creating a linear coordinate system that can be used for navigation and exploration. Our main ideas for this representation are ideas of abstraction at the block level, summarization, since we provide information on the pangenome rather than the individual genomes, and interactivity for the exploration navigation. It is available as a JavaScript application and is quite similar to a genome browser. On the top right, there is a miniature for navigation that can provide summarization information for the current chromosome in view. Next to it, there is an annotation track that can display annotation cards for the genes, if you have any. And then there is a presence-absence matrix with different tracks of information for the pangenome. On the left, there are multiple customization options. Here you have the chromosome choice to change the current chromosome in view. You can choose the threshold that should be applied for the definition of the core genome, choose the zoom level at which to see the block in display, and other options such as the hollow area finder that can auto-detect consecutive absences. Let me introduce some of the most interesting functionalities of Panash. First, the core genome threshold. Within Panash, the core or dispensable status of each block is displayed on an additional track below the presence-absence matrix. Blocks in orange are belonging to the core genome, while blocks in blue are belonging to the dispensable genome. However, you can choose to change this value of threshold, and then the color will be applied accordingly directly on the visualization. We also added an option for the uploading of GFF file of annotation. Within those files, the genes annotation will be extracted and laid out on a dedicated track on top of the presence-absence matrix. By hovering the marks corresponding to the genes, you can show a tooltip or an annotation card with information on the gene you have been selecting. Last but not least are the recently added sorting options. You can now choose to sort the presence-absence matrix depending on different criteria. I won't be detailing all of them here, but you are welcome to ask me any question at the poster session or outside of it. For example, you can choose the local presence-absence pattern option. This sorting option will calculate a local phylogeny depending on the motifs or patterns of presence-absence for each of the genomes and order the genomes accordingly within the matrix. I will conclude by saying that Panash is available on github.com as SouthGreen platform Panash, with more documentation and demo versions available. You're also very welcome to come to me and ask me questions. I'll answer them gladly. Thank you very much.